In the last lecture, we spoke about the enzyme and its nature. That was the first part of the enzyme. Let's talk about in the same series, the enzyme part 2. A very warm welcome from Spice Up Your Biology. This is Sandeep here. A warm welcome to all of you. Hopefully, you will like this. Watch till very end. Subscribe and share my uh, channel. It will be a great pleasure to present you many more such content. Let's go about it. Just I give you the brief. In the last lecture, we spoke about how exactly enzyme function. So what I told you in that lecture is that the enzyme considering a big molecule and the substrate is going to be very small. A very small reason, a fraction of the enzyme part, because the active site or the catalytic center, which governs the all the activity that is happening between the enzyme and the substrate. Enzyme is going to hasten it up by lowering the activation energy. This is done just the bringing certain goods together which are going to react together. Otherwise, if they collide each other, it will take time. That is how the enzyme is going to increase the rate of reaction, and not one or two times. It's going to be in the range of around 10 power 6 to 10 power 12, and sometimes even more. So let's talk about further. I will just talk about the cofactor. So cofactor, as I told you, is the umbrella word, and one of them is going to be the coenzyme. And this coenzyme is a kind of organic molecule which will always associate with the enzyme with the non-covalent linkages, especially the weak interactions, maybe the hydrogen bond or maybe some other bonds involved, which is not covalent in nature. And many of those coenzymes, they're known to be uh, vitamins. You can see the certain category of the vitamins, and they play a very specific role. For example, vitamin B1, that is the thiamine pyrophosphate. It is always involved in the decarboxylation reaction, whereas the riboflavin, vitamin B2, is FMN and FAD, which is going to be formed from there. They work like a carrier of H plus ion. Then we have the niacin, that is the vitamin B3. We have the NAD and NADP plus, which is a carrier of hydride ion. Then we have the B6 pyridoxine, basically the pyridoxyl phosphate and pyridoxamine phosphate. They're the carrier of the amino and the carboxylic group. Similarly, vitamin B12, that is the deoxyadenosine cobalamin, which is a coenzyme in the amino acid metabolism. Folic acid, that is actually in the form of tetrahydrofolic acid. So they always work as a coenzyme when the one carbon transfer is going to be there. Pantothenic acid is a coenzyme A, acyl group transfer. Biotin, which is a coenzyme in the CO2 fixation. Ascorbic acid, they do play important role in the hydroxylation of the proline and the lysine residues, which form the triple helix. So strength of those collagen proteins are given by the this ascorbic acid, which strengthens up the muscles. And there's a certain deficiency diseases which is going to happen. Before I go into that, I tell you the cofactor. It could be of two types. It could be the metal ions, or it could be coenzyme. In coenzyme, it could be the co-substrate, or it could be the prostatic group. Prostatic group is one which is covalently bound, and co-substrate is one which is loosely attached. This is the current way of defining the cofactors. Let's talk about the certain deficiency disease. For example, the cobalamin or vitamin B12. This causes the pernicious anemia, a kind of anemia which is hidden. Folic acid, if it is not there, this will cause the megaloblastic anemia. Nicotinamide will cause the pellagra. And similarly, the riboflavin or the thiamine, they call the, especially thiamine, they cause the beriberi. Now, if you see the structure of the NAD, it can exist in the two form, NAD plus, which is the oxidized form, and NADH, which is the reduced form. Reduced form, it is going to give off the energy. It is equivalent to the 3 ATP. If you look at the structure, why it is called so, nicotinamide adenine dinucleotide. If you look at the structure, this gives you the basic part, which is of the adenine residue, or you can see the adenine and the one phosphate group, and there's another the group which is the nicotinamide over here, and again here the sugar is going to be involved. So we have this is the region is called a dinucleotide, and this is minus ion hydride ion will be taken over here. So they derived from the vitamin B3. NAD plus to NADH mechanism. I told you that NADH is going to be energy rich compound, and this is how it is going to be protonated. You can see over here. This is the oxidized form, and this is the reduced form. Now, another adenine dinucleotide coenzyme, that is NADP. You can see over here, this ribose is going to be phosphorylated over here. And the FAD, that is a flavin adenine dinucleotide, you can see over here. So adenine, and you can see the structure which is of the flavin. Now, the certain environmental factor, this I just described in the last lecture in very brief, is that about the structure of the enzyme, especially in the active site, is going to be affected, under the certain environmental factors. For example, if the pH is going to, all the enzyme has a pH optima, which is in the most of the cases going to be in the neutral range, but it can be enzyme specific as well. For example, the enzymes which are found in the lysosomes will be active at the acidic pH. And some other conditions, enzyme may be active at the alkaline pH as well. Now, what is happening if you are decreasing or increasing the pH, rate of reaction is going to be highly affected. So it will show its maximum activity at the optimum pH of that particular enzyme. The explanation can be that is that uh, the pH is one which maintains the active site. So if the active site is going to be distorted, or the protein structure is going to be distorted at a particular pH, it will not be functioning in the way it has to be. So that is the basic reason. Certain enzymes I told you, other than that, the pepsin, chymotrypsin, pepsin works at the very low pH between 1 to 3, and the chymotrypsin is going to work, the pH above 8. So they have a different absorb, the pH optima. That is very important for its functioning. Then we have the temperature effect. If you see the uncatalyzed reaction, so the rate of reaction will probably know about the temperature quotient, where we increase the temperature from 25 to 35, the difference in the 10 degree, it doubles the rate of reaction. So if you see here, the uncatalyzed reaction, the temperature is a factor which will increase the rate of reaction. Whereas in the case of enzyme, it is again going to give the bell shape. Every enzyme has its optimum temperature, where it shows it's the maximum activity or optimal activity. Same region, like if you're increasing or decreasing the temperature, that will disturb the protein structure, and hence the activity will not be there. So excessive temperature or the excessive heat can denature the protein. So important hydrophobic interactions is going to be disturbed. In that way, they will not be able to function the way it was doing. Now, there will be certain ways regulation of the enzyme activity. The few things which is very, very important. Before I go into that, I tell you when you talk about the catalytic efficiency of the enzyme, so you need to be dependent on the two factor. One is a Km value, that is a Michaelis-Menten equation. More is the Km value, less is going to be the affinity of enzyme towards that substrate. If the one enzyme which is having the two substrate, so one substrate will be much dearer to it than the other, which has the less Km value. That means the Km value more denotes the uh, less affinity. The second factor is a Kcat, that is a turnover number. So whenever you going to see the enzyme, so the ratio of the Kcat and the Km value, this ratio gives that the enzyme is very, very efficient. So any enzyme which is having this ratio Kcat by Km more, that means that enzyme is more efficient than the other one. Now come to the point. There are many ways in the biological system the enzymes are going to be regulated. First, they produce enzyme only when the substrate is present, which is common in bacteria. This is, a kind of induction or the repression, or enzyme system can be inducible, or system can be the repressible. Then the allosteric enzyme, where we have the inhibitors or the moderators, which can alter the rate of reaction. Then we have the feedback inhibition, where the product is going to bind to the enzyme and give a signal that enough product is formed, and hence the enzyme reaction is going to slow down and eventually stop. Then we have the zymogens. These are the enzymes which are not in the active form. They need to undergo the catalytic cleavage, and then only they'll be active. For example, the trypsinogen, pepsinogen, chymotrypsinogen, all they need to be cut in order to get function. This is how the cell maintain the high enzyme level, but it will be active only when the substrates will come and the certain signals will come, and then it is going to be cleaved and the process to get activated. Then we have the protein modification. The certain modifications, especially the phosphorylation, dephosphorylation, which matters a lot in order to activate or deactivate the protein. Let's first talk about the allosteric enzyme. Allo means a different. So here the steric means position. So here uh, the moderators, which could be the activator or which could be inhibitor, will bind to the other place. So this, as I told you in the beginning in the last lecture as well, uh, if any enzyme is allosteric enzyme, it will never follow the Michaelis-Menten equation. Rather, it will follow the sigmoidal curve, which is of the monad kinetics. The second thing is that here the effector molecule, that could be the positive or it could be the negative, will bind to the other place. Other place means other than the substrate binding site. So there'll be two conditions. One is a relaxed, another one is a tight. So if any enzyme, allosteric enzyme in the tight condition, will not have the enough activity, and when it is in the relaxed form, it will have the more activity. So positive allosterism will always increase the relaxed form, and whereas on the other hand, the negative allosteric or the inhibitors, they will always make the protein to stay more in the tight conformation. So these conformational chains are very, very important for the activity to take place. So the few enzymes, one is the fructose 6 phosphate. You can see over here, which carries out the phosphorylation of the fructose 6 phosphate to get converted into the fructose 1,6 bisphosphate. Here the negative factor is going to be AMP, and sorry, ATP is going to be negative factor because already the enough energy is there in the system. So why to make more? Whereas the AMP, that is a signal that the energy level has been reduced, so that is going to work like a positive effector, and hence the rate of reaction will increase. Feedback inhibition, as I told you, like suppose a product, a substrate, it begins with the A, and the final product is going to be F. So the finally F will come and bind to the E1, and hence the rate of reaction is going to be stopped. So this is a kind of feedback inhibition which some of the enzymes undergoes. And the proenzyme or the zymogen, I told you before as well, these are inactive form. They need to be converted to the active form by the hydrolysis or the autolytic proteolytic cleavage. And there are many enzymes in the digestive tract which needs to be cut. For example, the proelastase, trypsinogen, chymotrypsinogen A, pepsinogen, and procarboxypeptidase. The proelastase and trypsinogen, they are going to be activated by the trypsin. Chymotrypsinogen A is going to be cut by the trypsin and chymotrypsin, and the pepsinogen, which is required acidic pH and a little amount of the pepsin, and the procarboxypeptidase, they need to be cut by the trypsin. Now the protein modification, there'll be two kind of modification which can occur in the protein. One we call as a O phosphorylation, another one is a N phosphorylation. O phosphorylation usually occurs on the amino acid which are containing the OH group. The three amino acid you can put together, serine, threonine, and the tyrosine. They need to be phosphorylated in order to get active or in order to get inactivated. So phosphorylation and dephosphorylation can work either way. They can activate a protein or they can deactivate the protein. And the certain inhibitors, there'll be uh, different category of the inhibitors. They play important role in the drug discovery and all. So one is a irreversible inhibitor, which bind enzyme with the tightly and prevent the formation of the ES complex. This is not going to play any role in the enzyme kinetics, whereas the reversible, which could be competitive, which can be non-competitive, which can be uncompetitive, which plays a very important role in regulating the enzyme and which is being used to study the enzyme kinetics. And irreversible, as I told you, that the certain one, like for example, the arsenic, they have the snake venoms, which works on the many peptide. Arsenic, they work on the certain important enzyme of the Krebs cycle. Nerve gas, which is also going to work on the certain neurotransmitters. So they are going to be irreversible inhibitors. Reversible, as I told you, they will bind. They will not bind, to the enzyme permanently. So they can be removed. So their binding is going to be reversible in nature. That can be competitive, they can be non-competitive, they can be uncompetitive, depending upon like if the binding site of the substrate and the inhibitors, they're the same. So this kind of inhibition we call as a competitive inhibitors. So apparently there'll be change in the Km value. We'll discuss that is a long thing. And inhibition can be overcome by the increasing the substrate concentration. So you can see diagrammatically, this is the normal substrate and this is the one competitive inhibitor which is binding, So this will block the reaction, slow down the reaction. And if you see the non-competitive, reversible non-competitive enzyme inhibitor bind to the R group of the amino acids or the metal ion cofactor, and this binding is usually weak. Enzyme activity is restored when the inhibitor dissociate from the enzyme inhibitor complexes. So they do not bind to the active site unlike the competitive one, and they do modify the shape of the active site once bound elsewhere the structure. And the certain other thing I told you, the proteolytic enzymes. Proteolytic enzymes cleave the peptide bond in order to make the other enzyme active. So this is going to be, the enzyme which can work on the C terminus or they can work on the N terminus of the protein. Now, if you see the specificity of the proteolytic enzyme, for example, the chymotrypsin, there are the few chymotrypsin. There could be they belong to the hydrolysis only. It could be the chymotrypsin, it could be the trypsin, it could be last stages. So pepsin can be another one. You can see what matters over here is the catalytic domain. Some of this enzyme they work on the C terminus, other they work on the N terminus. And secondly, the choice of the amino acid that varies. Some they look for the hydrophobic small amino acids, some will look for the aromatic amino acids, bigger in the size. For example, the tryptophan, tyrosine, or the phenylalanine, or other just look for the positively charged amino acid. So that is very important because most of these enzymes are the member of the serine proteases, and they have a kind of catalytic domain where their desirable structure of the substrate will fit in order to carry out the reaction. Now, if you just look at the evolution, so pancreatic serine proteases are a category of the enzyme which all hydrolyze the peptide bond. So they appear to have arisen from the divergent evolution from a common ancestor. So they have the similar primary structure, similar tertiary structure, and different specificities as well. Each enzyme has a pocket which is different depending upon their substrate molecule, and so that they can fit in. You can see the chymotrypsin and the elastigate, they were shown over here in this diagram. So chymotrypsin will always go for the carbonyl side of the C terminus, and they look for the aromatic amino acids which are large and hydrophobic amino acids. Trypsin is one which also work on the C terminus of the basic amino acid, which is lysine and arginine, and elastases will look again on the C terminus. They look for the small hydrophobic amino acids like the glycine and alanine. There are certain uses of the enzymes in the medicines and the diagnostics. For example, the when we look at the level of those enzymes during the heart attack, LDH, creatine phosphate, SGOT, if the pancreatitis occur, it is the inflammation of the pancreas, amylase and lipase levels are going to be checked, and certain analytical reagent which is going to use them, so that they can be analyzed and they can be measure the substrate level. For example, urea converted to the ammonia via the urease. So blood urea nitrogen is going to be measured. Replacement therapy, administer genetically engineered beta glucocerebrosidase for the Gaucher disease. So there'll be many ways the enzymes can be used, and they are, the many of the enzymes, they are the part of the the drug discoveries, or the many drugs they are going to tag them one way or the other to find out the solution for the certain disease. So that's all for today. Hopefully you like this lecture. Thanks a lot for hearing me. Thank you.